Okay, so in the last video, we did replication. Replication is where we start with DNA, and we're going to end with DNA. This video, we're going to talk a little bit about transcription. Transcription is going to be the next step, and you're going to start with DNA, and you're going to end with mRNA. In the last video, we're going to talk about translation. In translation, you start with mRNA, and you're going to end with a protein. Okay, so replication, we did, that's the last one. This is where we're going to start. Okay, so as a review, we are going to talk about nucleotides. Remember, nucleotides have three parts. You have a pentose sugar. Okay. In DNA, that's a deoxyribose. In RNA, it's a ribose sugar. Then we have a nitrogen base. In DNA, remember, we have A, which always binds with T. We have C, which always binds with G. Remember, those are Chargaff's rules. And then that last part, so we have the sugar, the base, and then we have a phosphate group. Now, when we talked about DNA, we said that DNA is always going to be double-stranded. Okay, it is in the form of a staircase or a twisted helix. Okay, and your sugar is going to bind with the phosphate group of the neighbor nucleotide, and that forms your backbone. And we talked about how one strand of your DNA ran 5' to 3', and the opposite strand ran 3' to 5'. And we talked about how replication was a semi-conservative method of replication. Okay, so you have all of that down. Now we're going to talk about RNA and how we get from DNA to RNA. So DNA is located in your cells. And where is it located in your cells? In your nucleus. Can DNA get out of the nucleus? No, it has to stay in your nucleus. It is safe and protected in the nucleus. But if DNA codes for your proteins, how does it get out? So the code is going to come out of your nucleus in the form of mRNA. So we have three types of RNA. So RNA, monomer, is a nucleotide. Remember, a nucleotide has three parts. It has a sugar, a pentose sugar, it has a phosphate group, and it has a nitrogen base. Now for DNA, remember it was ATCG. For RNA, it's a little different. You have A, you have C, you have G, but A is going to bind with U. U is uracil. Okay? There is no T in RNA. It's replaced with a U. Now we have three different types of RNA. We have messenger RNA. We have transfer RNA. And we have ribosomal RNA. Okay, we're going to abbreviate these as mRNA, tRNA, and rRNA. mRNA. Okay, there are some other kinds of RNA, but we're not going to talk about those in this class. And for this lesson, we're going to talk mostly about this one right here, our mRNA. Okay, so we have our cell. Okay, we have a phospholipid bilayer. That makes up our cell membrane. Inside we have our nucleus. We have endoplasmic reticulum. We have ribosomes. Little dots are our ribosomes. We have our mitochondria, which is a powerhouse. Okay. You have your centrioles that we talked about when we did mitosis. Okay. You might have a vacuole. Okay, all of that stuff, but we're focusing right here inside our nucleus. Inside the nucleus, you have your DNA. Remember, your DNA cannot leave the nucleus. It's going to stay there. In order to get it out, to get to that ER, to get to those ribosomes, to make it into a protein, we're going to have to transcribe it with a C, transcribe it into mRNA. So we are going to make sure we go in order here. This is our DNA. Same DNA we had in the last video. We have DNA. Then we have these up here. Okay. See how these are different? How are these nucleotides different than these? They still have letters on them. The white ones have a U on them. Red still have the A. Green still have the G. Here's a blue one. Still has the C. Okay, but look at the shape. How are they different than these? Okay, now we're going to show you how it goes through. So just like last time, we have an enzyme. If I can get back far enough so you can see the whole enzyme here. So this is RNA polymerase. Okay, pretty big. I can't zoom out any more than that. Okay, RNA polymerase. You can see it down here at the bottom, RNA polymerase. So our DNA is going to come in on this side, and then it's going to split right here at the front. What does that remind you of? It's the same kind of form as our helicase, but this is RNA polymerase, so it's going to come up through here. Now, as it comes through here, your DNA nucleotide is going to come up right to this active site. This is your active site, okay? But we aren't binding DNA, we're going to bind RNA. So what binds with A? U. It's going to move to the next one. What binds with G? C. Okay. What binds with A? U. Now you'll notice that right after it binds here, it's going to unbind. Okay, RNA does not stay attached to DNA. So after it goes through that active site, it's going to unattach itself from the DNA. It binds just for a minute so that you can see and that they can see that it's the right match. Okay? So it's going to keep being pushed through here, binding for a second and then unbinding. Okay? G. C. See how they're still complementary here, but we're binding with RNA instead of DNA. A. bind there, and then a C, because it has to match up with there, and then another C. Okay, and what binds with A? U, because there is no T. Okay, so it's going to bind there. Now we're going to come over here to this side. Okay, so this is your RNA, your messenger RNA. It's binding, okay, matching up, complementary base pair here, and then your RNA is going to leave your polymerase. This is an mRNA strand. Your DNA, as it comes out the other side, is going to rebind together. Okay, it's only separated inside the RNA polymerase. Then, when it's done, when it reaches that stop codon, it's going to release your mRNA strand. This mRNA strand can now leave the nucleus. DNA does not leave the nucleus, but this mRNA strand that we just made can leave the nucleus. Okay, so it's going to leave the nucleus, it's going to go to the endoplasmic reticulum, and it's going to find a ribosome. This right here, with this RNA polymerase, our red mat, is going to be transcription. You have DNA coming in on this side. It's going to separate out. It's going to come up to this active site. Your active site is going to bind with an mRNA to form an mRNA strand, and then as your DNA comes out the other side, it's going to connect back together. When your DNA is done transcribing, your DNA goes all the way back together, and then this mRNA can leave the nucleus. mRNA is a complement to your DNA strand.